Thank you everyone so much for being a part of the webinar today, and thank you so much for participating in our overall webinar series. I'm Emily Boyd Stormoen, the CEO of the Arthritis National Research Foundation, and on behalf of our Board of Directors, our Scientific Advisory Board and staff, I'd like to welcome you all to the Spotlight on Osteoarthritis. We're excited to have so many of you joining us from all over the world, especially on World Arthritis Day, so thank you for your support. I'd also like to welcome our moderator for the discussion, Dr. Anne-Marie Malfait. Dr. Malfait is a professor of internal medicine and the Dr. George W. Stuppy Chair of Arthritis at Rush University. Dr. Malfait is also a member of our ANRF Scientific Advisory Board. Thank you, Dr. Malfait, for moderating this webinar. Before we get started, there are a couple pieces of logistical information we want to share. First, the webinar is being recorded and we'll have it available on our website and YouTube channel in the coming weeks. If you have questions, please type them into the Q&A, and if we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. Now, let's get started with today's webinar and hear more from our ANRF scholars. Dr. Malfait, I'll turn it over to you.
Thank you, Emily. I cannot start my video, so maybe this is in a locked mode. So if somebody could unlock that so you can see my face, that would be wonderful. I think it would be nice if we could see all the participants actually in the panel. You are now muted.
I'm sorry, please go ahead and we'll work on that while we get started with your introduction.
Okay, thank you. Well, so good afternoon, everybody. As you just heard from Emily, my name is Anne-Marie Malfait, and I am currently in Chicago, and I'm very happy to welcome the outstanding ANRF scholars here today, indeed on World Arthritis Day, which is there I am. Good afternoon. So what would be a better way to honor World Arthritis Day than a scientific seminar that highlights some exciting research on osteoarthritis. So osteoarthritis is actually the most common form of arthritis by far in the world. And so we have some very interesting research going on that is sponsored by the ANRF. And we will start with Dr. Jonathan Brunger, who is coming to us from Vanderbilt in Nashville, Tennessee. And so Dr. Brunger has been an ANRF scholar since 2021, I believe, and his area of focus is synthetically regulated cell-based therapeutics for targeted articular cartilage regenerative medicine. And so he will be talking to us about a new way of looking at stem cells. Dr. Brunger.
Good morning. Thank you for the introduction, Anne-Marie. It's fantastic to be here presenting to this group. Today, my talk is on stem cells and osteoarthritis, and I want to begin by talking about what is osteoarthritis. So osteoarthritis is characterized by joint space narrowing, and you can see this in a radiograph comparing an arthritic to a healthy joint. This is a healthy knee, where there's nice joint space preserved here, whereas it's lost in the arthritic knee. We know that in osteoarthritis, a variety of joint structures actually undergo remodeling, as illustrated in this cartoon comparing normal knee tissue to an osteoarthritic knee, where we see damage to the articular cartilage and remodeling of this underlying bone, and also inflammation of the synovium that encapsulates the joint. Osteoarthritis occurs for a variety of reasons, including physical damage through trauma, the onset of metabolic diseases, genetic factors, and ultimately these different features can come together and interact with one another, and they lead to a situation where there's a high degree of inflammation in the joint. This is what gives rise to degradation of this lubricating tissue, the cartilage in the joint, that is a hallmark of osteoarthritis. Now, as with OA having different causes, there are also a variety of treatment options available to patients at different stages of the disease. These can be lifestyle modifications that allow for better management of the disease. some injections to lead to relief of short-term relief of some of the symptoms of the disease, but these don't really reverse or modify the course of the disease in any positive way. Biologic treatments that can slow the progression of the disease, but don't really provide a long-term solution. And ultimately, at the end stage of the disease, often osteoarthritis is treated with joint arthroplasty, which is the replacement of the viable native tissue with hardware in the joint. Each of these options can be fantastic depending on the disease state, but none of them really provide long-term therapeutic treatment that is disease modifying. That is to say, allows the joint to be restored to its native health. And even the replacement of the tissue actually can result in some problems over the long run if these joints ever need to be replaced, which is occurring more and more often as patients need joint replacements earlier in their lifetimes with increased lifespans. And so we're interested in developing technologies that can either eliminate the need for these hardware transplantations or at least delay them in a meaningful way. So we're interested in stem cell technology. Why are we interested in stem cells? Well, because they can self-renew themselves. They can produce more of their own kinds of cell, and they're highly plastic. That means that if cultured or maintained in the right conditions, we can actually coax them to take on a variety of functions. Functions as diverse as becoming cells that reside in bone, cells that can later become neurons, or of course, as we'll talk about, cells that can even become cartilage tissue. Furthermore, some classes of stem cells are thought to produce factors that can promote homeostasis. That is to say that they can interact with other cell types in different tissues in order to help the tissue maintain a healthy, normal function, even in the face of inflammation that can drive diseases such as arthritis. So that's why we're interested in stem cells. Now, a little bit of a caution. Not all stem cells are equal. And you might see, even driving down the road, or when you visit a physician, signs that are advertising stem cell therapeutics. And I just want to caution you that although I'm super enthusiastic about stem cells and cell-based therapies, I do not think of all of these things as offering the same kind of promise. So not all stem cells are equal. And as an example of this, I'll just highlight a published clinical case where one patient who was participating in what's referred to as stem cell tourism, so really seeking to have cell-based therapies for a disease that was ailing this patient, actually went around to numerous clinics and received transplantation of stem cells. And this resulted in a massive problem for this patient that was trying to treat his ischemic stroke, ultimately ended up with a mass growing along his spine. And you can see this here, both in this MRI image of the spine, as well as when the surgeons needed to go in and try to remove this tissue. And when we look at this tissue, what is occupying it, we see markers of cells that are causing problems for this patient and growing in an uncontrolled fashion. This is not really a cancerous tumor, but it is a lesion causing an additional problem for this patient, introduced as a consequence of really highly unregulated cellular transplantation therapy. What we're interested in is think about stem cells and bringing the power that they have in biology to bear for us in controlled fashions for patients. So if you see here, this is actually a developing mouse embryo. Each dot represents a cell. And I don't want to spend a lot of time on what this is, but I do want you to appreciate that even in this developing embryo, these cells are working together in a highly coordinated fashion in 3 different developing embryos to form these different structures that are later going to give rise to a living mouse. And we all know what a mouse looks like. And we can see that biology is capable of reproducing this over and over and over again through these coordinated features. We think of this as something like programmable robots, where robots can be programmed to even play team sports together. Although I hope you'll notice here that the action is not all that thrilling, and even the performance of the participants leaves a little bit to be wanting. What we're interested in my lab is thinking about cells as therapeutic agents that we can program. We actually know a lot about how cells receive information, process it, and provide feedback to their own environments. And what we want to do is implement cell engineering strategies to allow this to be done in a reproducible fashion. So we know what cells are going to do when we transplant them and how they're going to do it, what kind of decisions they're going to make, and what kind of outcome that might have on tissues down the line. As we apply this kind of approach to osteoarthritis, what we would really love to do is have a cell that can literally, through a programmed recognition motif, recognize the arthritic lesion in the damaged cartilage, and then implement some sort of programmed function that is going to allow the tissue to repair itself. So what we've done is we've taken synthetic receptor signaling modules where we are literally able to program how cells read and react to the contents of their environments. We can program them through building receptors to recognize things like degraded extracellular matrix, which is the damaged cartilage, and implement a variety of responses that might be instructive for producing healthy native tissue in a damaged joint. So with the goal of really elevating the play of these robots, cells, in other words, stem cells, in order to be effective agents of therapy. So a graduate student named Bonnie Walton in my lab has been working on this technology for a few years now, and we're happy to report that we can build cells that can exquisitely recognize particles that are signatures of damaged cartilage. And we can detect it at very low concentrations in models of cell culture. And this is just showing these different concentrations here are just showing a differential response to a variety of levels of the molecules that the cells we're engineering to detect in damaged tissue can be detected. And we even get elevated responses up to a point. We also know that these cells are specific to the type 2 collagen that is exposed in damaged cartilage in arthritic conditions, not type 1 cartilage that is found in other tissues. So we're seeing that our receptors can specifically and uniquely respond to features of damaged cartilage and not other physiological features that might be encountered by the cell that is kind of introduced to and crawling around in the joint. We then took these engineered cells and placed them on damaged cartilage in a chamber where cells can also crawl off of the cartilage. And in this way, these cells are engineered to produce a kind of color that we can detect through our microscope. And so this dashed region represents where the cells are growing on this cartilage matrix right here. And then inside of the well itself, off of the cartilage matrix. All of our cells here that are programmed with a receptor are blue, but the cells that are on the cartilage matrix, recognizing the physically damaged cartilage that we sliced with the blade, they're actually turning red as a consequence of the activity of our artificial gene circuit. And so you can see here that we have this high spatial control that is provided through this artificial rewiring, this synthetic signaling, where our cells are actually uniquely capable of recognizing damaged cartilage instead of just, their own native environment. So the question became, can we actually program these cells to do something other than glow interesting and beautiful colors? And the answer is yes. And we think this provides the opportunity to localize therapies of drugs. And so just to illustrate a few key points, we can cause cells to produce molecules that might antagonize inflammation or promote new cartilage synthesis that other studies that we've done have already shown allow us to build productive cartilage that can sustain the inflammation signaling that is typical of osteoarthritis. So just to wrap up real quickly, we have a combination of stem cell technology and tissue engineering and what we call synthetic biology, these artificial receptors, where we're building cells as reliable therapeutic agents to treat arthritis. They intelligently survey their surroundings before implementing a regenerative program that is auto-regulated and based on the signature of disease, arthritis. And we're happy to say we're performing ongoing work where we're testing this cell design strategy in experimental models of arthritis. With that, I thank you for your attention. I acknowledge my lab members, our collaborators, and our funding agencies wanting to highlight the important work of the Arthritis National Research Foundation. Thank you so much, and I'd be happy to take any questions.
So while we are waiting for questions, I have a question for you. I've now heard this several times and in bigger detail. So I think this is really exciting. Why are you are not really limited to doing this in cartilage, right? You could do your system. I'm asking within the joint here now, not just outside the joint, but you could engineer cells that in the synovium or anywhere you wanted, correct?
That is certainly follow-up work that we're planning to pursue. We are interested not only in doing this in stem cells, but in other cells that you find native to the joint, cells that are in the synovium, for example, but also cells that infiltrate to the joint that might not reside in the synovium too, that might be present in the synovial fluid. So you're absolutely right. We're not restricted to a particular cell type or even to the joint space or a tissue that is associated with the joint. We're deploying this in the central nervous system for neurodegenerative diseases that are also associated with aging. So we're very excited about applying this technology far and wide.
Yeah, it is very attractive technology. Are there any questions from the audience, please, or from any of the other panelists?
Yeah, one other question that came in, Dr. Brunger's, in the future, if this is able to be used for patients who are suffering from osteoarthritis, how long does the regeneration take place once the stem cells are placed in the affected area? Would it be instantaneous, over a couple of months, a year? When would someone expect to see results?
That's a great question. So there are really two angles of attack we're taking here. The first is to try to stop the deterioration, and the second is to promote restoration. Promoting restoration is not something that has been done very successfully in the joint. So it's actually very difficult to speculate as to how long that will take. But we believe that it would take on the order of weeks to months to begin to obtain productive neotissue that looks like the tissue that typically resides on the cartilage surface. But in terms of abating the inflammation that drives the progression of the disease, we think we can do that quite quickly. So first, stop further deterioration and then promote restoration.
Okay.
Can I ask you a question, Jonathan?
Of course.
Hi, Jonathan. Really great work. So I'm just wondering, like, if you promote the chondrogenesis from stem cells to chondrocytes, is there a way, we know that chondrocytes can also terminal differentiate into hypertrophic chondrocytes, which, they actually not very helpful. Is there a way you can prevent that using your engineering strategy?
Potentially. So part of it is going to depend on what sort of molecule we're having the cells express when our circuit is activated. So if we are very careful about selecting that, then we think we might be able to circumvent this problem to some degree. In terms of actually having the chondrocytes be the modified cell, that is something that we're looking at in the future, but that circuit design is going to have to be very different just because of the way the engineered input is perceived by the cell. It's a nuance of the platform we're using. I'm happy to dive deep into that, but this might not be the right forum. So we think that there are ways that we can rewire what the chondrocyte is detecting and then dictate what its output is going to be on the basis of that. And that might promote chondrocyte function independent of the sort of circuit I described today.
Thank you.
Thank you, Jonathan. One quick question. So which models are you testing, or what is your first choice model that you will test this in vivo?
We have ongoing experiments where we're testing this in a mechanically loaded model of osteoarthritis. It's really something of a post-traumatic osteoarthritis model. So in this model, the joint is loaded quite severely over time so that the joint develops arthritis. So we have studies ongoing right now where we're doing that. We think probably the second iteration of this is going to involve what might be a better model for the sort of disease that we're expecting our cells to do well in. And that would be the ligament tear model, similar to what Ben Christensen is working on. So that's what we're anticipating.
So this is rodents that you're doing this?
Oh, I'm sorry. Yes, this is in mice. Yes.
Okay. Thank you. Thank you very much, Jonathan. And we will go now to the next speaker. So the next speaker is Dr. Charles Chan, who is an assistant professor of surgery in the plastic and reconstructive surgery department at Stanford University. And Dr. Chan is a first-year recipient of an ANRF award, and he also has a new project that focuses on skeletal stem cells. Now, unfortunately, Dr. Chan last minute could not be present here today, and he has been so kind as to tape his recording for us. And so that is what we will be listening to now. Thank you.
Just getting that pulled up and just be a second longer. It looks like James is pulling it up on his computer. So James, if you can share that again with us, that would be great.
I'm an assistant professor at the Department of Surgery, Stanford Immunology, and at the Stem Cell Institute. I first want to thank
we cannot see it, James.
Readers, friends, and my colleagues.
Sorry about that, James, but we could not.
The title of my talk is Skeletal Stem Cells and Beyond. Our group uses skeletal stem cells as a platform for understanding important questions such as how are we shaped the way we are? How do we stay this way? And of course, can we reverse skeletal diseases such as osteoarthritis? Osteoarthritis afflicts over 78.4 million adults in the US each year. And the disease manifests itself as cartilage damage, along with signs of new skeletal formation, as seen by these formation of these osteophytes here. But the goal of our research group is to try and find out the mechanisms for the development of this disease and ask whether or not skeletal-genic stem cells could be used to perhaps regrow cartilage that has been lost. How do we find these types of skeletal-genic stem cells? We use a technique called fluorescence-activated cell sorting that is enabled by this machine. You can think of it as an advanced jelly bean sorter, which allows us to separate out cells based on expression of specific surface markers. Using this technique, we're able to go inside skeletal tissues and isolate the various cell types within it. For instance, you can see in this cross-section of the bone marrow that skeletal tissues are made of many different types of cells. Here are fat cells, some blood vessel cells, there are yellow staining osteoblasts, and there are many other types of fibroblastic as well as nervous and neuronal tissues. In order to see whether or not we are able to actually identify a type of stem cells, we first have to validate its function, ideally inside a living organisms. So we have devised an in vivo functional assay to test the activity of different types of stem cells that we isolate. After mechanically dissociating skeletal tissues, we use this machine to separate out different fluorescent antibody stained populations, which we then transplant into the kidney capsule of an immunodeficient mouse. If their cells are capable of forming any type of tissues, we would know because the donor of these cells are labeled with green fluorescent protein. Here, in this case, this particular population forms blood-carrying blood vessels. Here's another population that forms little fat cells. And finally, we were able to identify multiple populations that when transplanted gives formation of little bone ossicles. On occasion, we also find that there are cartilagenic tissues within these ossicles. So to summarize some of the work that we have done, using the technique of flow cytometry, we were able to identify specific types of self-renewing mouse skeletal stem cells that gives rise to a lineage of skeletogenic progenitors, which then in turn gives rise to mature bone, fibroblastic stroma, and cartilage tissues. We were able to isolate these skeletogenic stem cells in mice in 2015, and using this as a model, we also were able to identify the human skeletal stem cells in 2018. I also want to bring attention to the excellent work from our other colleagues in the field who have also subsequently identified other types of skeletal stem cells. One unique property about skeletal stem cells is that you're able to induce their activity and expansion through an injury response that might be triggered, for instance, by a surgical procedure called microfracture. In microfracture, the surgeon would induce small surgical lesions on the surface of a joint. This would then result in formation of a blood clot, and this blood clot would then be filled with what we call fibrocartilage. Now, this is not true cartilage. It's more of a scar type of tissues, but it's better than their previous condition of a damaged bone lesion in a joint. However, by mapping the lineage relationship between true skeletal stem cells, we were able to find that these fibrocartilage are also descended from the same stem cells that can give rise to both cartilage and bones. And by identifying other specific factors that control this process, such as BMP2 and VEGF, we were able to shift the activated skeletal stem cells from forming bone and cartilage, from forming bone and fibroblasts to forming strictly cartilage. So we were able to show that this occurs in mice. Here is a knee joint in mice that had been injured, but then transplanted with no factors. And here is a knee joint that has been transplanted with BMP2, which induced the regeneration of the tissues, but forming yellow staining bones. However, in this case, we have injured the tissue and then transplanted BMP2 and VEGF inhibitor, which now leads to formation of blue staining cartilage. We then showed that these combination of factors, BMP2 and VEGF inhibitor, also are able to form cartilage in human joint tissue that had been grown on the back of a mouse. And this work was published by my colleagues, Matthew Murphy and Lauren Kopke in the lab, in a recent manuscript in Nature Medicine, whereby we showed that by activating skeletal stem cells, we could regenerate cartilage in osteoarthritic knees. And this work we were also thankfully able to share with the broader community through an article in the New York Times and Forbes magazine. Now, what our group is hoping to do is we are hoping to try to scale this approach up further for use in larger animals and hopefully eventually leading to clinical trials in humans. Eri in Takematsu, a postdoc in the lab, has devised new methods of forming skeletal scaffolds, which could be transplanted into larger animals that can induce cartilage formation. Here, this is blue staining cartilage grown from these umbrella caps of cartilogenic scaffolds that releases factors that acts on resident skeletal stem cells, and that can cap like an umbrella on top of a damaged joint. More recently, we have started testing these scaffolds in larger animals, such as Yucatan pigs. By using MRI, we induce a defect in the knee joint of a pig. And there you see that with the application of these factors, you can regrow some of the lost cartilage. This is histology showing that in the absence of these factors, you would form fibrogenic fibrocartilage, which stains white. But with BMP2 and VEGF inhibitor, you form stable articular cartilage that has the mechanical properties of true cartilage. We're now using this approach and extending the technique further to see if we can regrow cartilage that has been lost in other areas, such as the craniofacial skeleton, to regrow cartilage in the ears, in the nose, and also in the temporomandibular joint, which is also affected by osteoarthritis. And you can see from these instances that there are blue staining cartilage that have been transplanted with just these cartilogenic scaffolds. So in summary, our team has identified skeletal stem cells in mice and humans. We have mapped the lineage relationships between their primitive self-renewing stem cells and mature progenitors of bone, cartilage, and fibroblast tissues. And this analysis identified new ways to control the specific formation of skeletal tissues by these skeletal stem cells, including BMP2 and VEGF inhibitors, which just, when applied to stem cells, skeletal stem cells, forms cartilage. Now we're working on new methods for delivering the stem cell acting signaling factors, and we're testing these cartilogenic scaffolds in larger animals. And this is my team who has done most of the work. And of course, we want to thank our funding sources, especially the Arthritis National Research Foundation. Thank you very much for your kind attention and your support.
Wonderful. So unfortunately, Dr. Chan cannot be here to answer questions. So I don't want to put you on the spot. This is clearly very exciting work by Jonathan. So could you maybe comment on this and maybe also for the non-scientists who are listening, put this into context and relate this to the work you just presented and that you are doing?
Absolutely, thank you. I think that work is very exciting. The trick is understanding the molecular bases for the performance of a cell. And what Dr. Chen has done very well is find the cell and understand what it is that's driving the performance of that cell, and then learning how to manipulate it in a reliable fashion. And so I think that approach has a long historical record in terms of other cell types being studied in the same way, and also has promise for utility in the clinic. Again, we can reproducibly understand what we're going to get when we use this kind of cell and provide this kind of like eluting cap that he referred to so nicely. And so I think that this has a complementary approach. The same mindset applies, governing the control for reliable expectations.
Exactly. Thank you for that. So that actually brings us back on time. So we will then move on, and I'm sure that Dr. Chan will be more than happy to answer questions from anyone. He was very disappointed that he couldn't be here today. So, and then we will go to our third osteoarthritis researcher, Dr. Shouan Zhu, who is joining us from the Ohio University, the Ohio Musculoskeletal and Neurological Institute. And Dr. Zhu is also a second-year ANRF recipient, and we will move away from the stem cells now. And Dr. Shouan Zhu studies a very important problem, which is metabolic dysfunction and its role in osteoarthritis. And I'm also happy to say that he actually had a paper accepted or published today or yesterday in the Journal of Bone and Mineral Research on high-fat diet-induced osteoarthritis in mice. It's a very interesting paper, and maybe we will hear a little bit about it today too. Shouan.
Thank you so much for the introduction. Good afternoon, everyone. So the overall goal of our research is to study why we develop osteoarthritis in our joints during aging and also during obesity. More specifically, we're interested in studying the role of metabolism is playing in this process. So as we know that osteoarthritis is actually a multifactorial disease, meaning that there are multiple risk factors can affect the disease development. Among those risk factors, aging and obesity are the two biggest ones. So if you're looking at the graph on the right, and this graph actually was generated from a national health survey interview that was conducted by the CDC in 2007 and 2008 on the US civilian population. And it's estimated that nearly between 10% to 20% of the US adults below the age of 50 have osteoarthritis, knee osteoarthritis. And this number actually increases every year as we age. And if you have obesity on top of this, you actually double this number. And this is true for both men and women. So obviously, there are many cellular processes can go wrong during this process, during aging. So in 2014, there's a prominent aging research group, aging researchers gathered together, try to summarize what's the common cellular process that's going wrong. So they came up with this so-called 7 pillars of aging, which includes the dysregulated proteostasis, stem cell exhaustion, and also our favorite metabolic dysfunctions. So the way we view metabolism, it's like the engine of the cars. Yes, it is important to have a good and new look for a car, but at the end of the day, it's really the engine that differentiates a good car and a not so good car. So another reason we're interested in studying metabolism is because it's playing a very important role during obesity and also obesity-associated other diseases, in this case, osteoarthritis development. Believe it or not, the cells in our joints, actually, they also have their own engines. So if we zoom into the joint capsules, you heard it from Jonathan, we have multiple tissues in the joints. We have articular cartilage on the joint surface, and also has a ligament connecting the tibia and femur. And if you zoom into the articular cartilage tissue, you see these round-shaped cells. These are the chondrocytes. And these chondrocytes have their own metabolic machineries that can generate ATP for their normal physiological functions. Unfortunately, this metabolic machinery is not working properly during OA. There's increasing evidence have shown that the mitochondria in chondrocytes have lower activities of electron transport chain complex 2 and complex 3. And there's also lower activities of glycolytic metabolism in chondrocytes during OA. So we're investigating why chondrocytes have dysregulated metabolism and trying to figure out if we can fix it. We know that metabolism is mediated by a series of enzymes. And these enzymes can actually be vulnerable to modifications and changes. And one of these changes is called protein myelination, or MAK. So this describes the enzymes. So in our enzymes, we have these lysine sites surround the protein. And these lysine sites can be masked by myelinol group and decrease its enzymatic activity, also decrease its metabolic functions. So the first experiment we did in our lab is to examine if MAK actually present in cartilage tissue, and if MAK actually change under different conditions. So we use a immunohistochemistry staining, as shown brown color in those pictures. We showed that there's a robust increase of MAK in cartilage during aging. And there's also an increase of MAK in cartilage during obesity. Then we want to figure out why this happens. It turns out in our cells, there is a metabolite called melanyl-CoA. And this melanyl-CoA can donate its melanyl group to the protein and lead to the upregulation of MAK. And the enzyme that makes melanyl-CoA is called acetyl-CoA carboxylase, or ACC. So ACC expression also increases in cartilage during aging, both the protein level and the mRNA level. And our cells actually have a very sophisticated system. It also has a defense system that can remove MAK from the protein and restore their enzymatic activity. And one of these proteins is called SIRF5. The bad news is SIRF5 actually decreases its expression in cartilage during aging. So this leads to our so-called two-hit model / hypothesis, is that, so we think SIRF5 decline in combination with ACC1 activation during aging and also during obesity, that together leads to upregulation of MAK and metabolic dysfunctions of chondrocytes that eventually lead to osteoarthritis development. Again, thinking about the car, we like to think that SIRF5 is actually a brake pedal for our car. It's actually not working as efficiently as we age, while the ACC1 is a gas pedal. It's keep pumping gas into the engine that eventually lead to the out of control of the car. So due to the time limits, I'm just going to briefly introduce what we have found, some of the results in this SIRF5 project. So in order to study the role of SIRF5 in osteoarthritis development, we obtained both wild-type mice and the SIRF5 knockout mice, meaning that in these mice, all the cells do not have SIRF5 gene. So we feed these mice either 60% fat, high-fat diet to induce obesity, or 10% low-fat diet as a control for 20 weeks. So we can keep track of their weight gain throughout the time. And you can see there's substantial weight gain in both wild-type mice and knockout mice. And then at the end of the study, we dissect the joints of these mice, and we cut the joints into thin sections, and we use a special staining protocol called saffron nose staining, and that allows us to visualize the cartilage tissue in orange and red color, and the bone tissue into green and blue color. So these are some of the results we have. So if you look at the first panel, you see the wild-type mice on the low-fat diet feeding condition, well maintained the joint structure, and the articular cartilage surface also quite smooth. There's no signs of degeneration. While the SIRF5 knockout mice, starting at this age, start to show some undulating joint surface, and there's also some places have loss of this red staining, indicating this joint is undergoing some early signs of degeneration. Under high-fat diet feeding, so wild-type mice start to develop some of these small lesions throughout the joint surface, while the SIRF5 knockout mice developed this bigger size defects, cartilage defect. So the results suggest that the SIRF5 deficiency in mice leads to more severe osteoarthritis development. We can also quantitatively characterize this phenotype by using two scoring systems. One is called OC score, the other is Mencken score. So we're also using very similar strategy to study the gas pedal, which is ACC. And currently, this project is still ongoing. With that, I'd like to thank my lab and also my collaborators in different institutes and universities, and also our funding from different places, especially ANRF. Thanks so much for your support.
Thank you, Shouan. I will open it up for questions, but while we are waiting for that, I would like to ask you a couple of very specific questions. So your staining with the MAC that goes up with age, and have you ever done that in meniscus? That was all mouse. I forget now. It was a human. Have you done it in meniscus also?
No. So the results here on the up panel, it's actually the human cartilage sample. We don't have the meniscus from a human, but we can certainly look at the meniscus in mice, either with low-fat diet or high-fat diet feeding.
And then can you remind me, so when you do your CERT5 knockouts at low and high-fat diet, how old were these mice? These were younger mice?
Yeah, so these mice we started at 6 weeks, and then we feed them for 20 weeks. Yeah.
And they were conventional, constitutional knockouts, right? Or it was not cartilage specific?
No, so these ones are not. So the cartilage specific knockout, we're actually in the process of doing that experiment because the breeding, took some time for us to get the right genotype. Yeah. We just started.
Okay, thank you. Are there any questions from the audience for Dr. Zhu?
I don't see any, but if you do have any questions, please feel free to type them into the chat, or you can always email us at info@curearthritis.org, and we'll make sure to get those to Dr. Zhu after the fact and get those questions answered. Very, very interesting work. And I think, as you said, Dr. Malfait, really fascinating to see the different approaches to treating osteoarthritis. And, I should mention all of our presenters today are published or recently published, which is quite a tribute to the work that they've done and the research that they continue to do.
Exactly. And it really highlights the how valuable the support of the ANRF is for the more junior researchers that we heard about today, and for the osteoarthritis field in general. I think all this work is truly fascinating, and I would like to thank all of you very much for participating today.
Yes, thank you so very much to our presenters and to our moderator, Dr. Anne-Marie Malfait. We're really pleased with all of our presentations and the work of our scholars. I think, as Dr. Malfait said, you can see firsthand the impact ANRF has in supporting these incredible individuals in their quest for a cure, especially as we celebrate World Arthritis Day today. Just want to thank all of you for joining us and for this great event. If you enjoyed this webinar, we kindly ask you to consider making a donation in honor of World Arthritis Day by visiting curearthritis.org/donate. Every contribution helps. Please remember to follow us on social media and stay connected. We'll have this presentation up and on our website in the coming weeks for those who were unable to listen and be able to connect with our scholars again. Thank you very much. Hope you all have a great day.